Hey everybody, how are y'all? Okay, I'm trying to get my iPad to mirror right now. 14 people enter the waiting room. How do I let everybody in? Hold on. Admit all. Okay. All right, looks like we got a lot today. All right, let's see. See if we can do this. Just trying to share my screen. Okay. Of course, it has to not want to work right now. We'll continue. Trust. If this works. Okay, I just admitted a few more people. Okay, can y'all see? Oh, not now. Okay, all right. So y'all can see my screen?
Yes, ma'am.
Yay. Okay, so a couple of people had sent a few things that they wanted reviewed. So I have on my list weak non-covalent interactions with hydrogen bonding. I have phospholipid flipping, DNA topology with a positive and negative, DNA twisting with a supercoiling with a positive and negative twist. Oh, let me admit some more people. I have Cdna Library, an application for next generation sequencing. Anybody else have anything else that we add to the list?
Mine is more of a general question, which is like, how should we expect proportionality of the chapter materials? Like
pretty equal.
Okay.
Pretty equal. Yeah. Okay, I'm admitting a few more people. Okay. All right, so anybody, let's see, I need to put in my chat. Anybody else have anything, any other topics they want reviewed? You could put it in the chat. I'm not gonna do this for more than an hour. I'm definitely gonna cut it off at that point, but you're welcome to hop in and out as much or as little as you want. So I'll start with the hydrogen bonding, which was, what was chapter 2, right? What was chapter 2? That's phospholipid. Let's see. So one of the questions was, what slide was that? Let's see, 34 here, was kind of just to review what is hydrogen bonding and how do you know what's the donor, what's the recipient, that kind of thing. Okay, and Tammy says Gibbs free energy calculations. So if you have one that you want to do, Tammy, if you get it ready, and we'll add that to the end of our list. But I didn't prepare any additional material for this. People just sent me, these are kind of my questions. So I'm happy to answer them, but like I don't have anything, I don't have an extra Gibbs free energy problem for you. Okay, so when we talk about hydrogen bonding, right? Hydrogen bonding is one of those weak interactions. When you think of a strong interaction, you're thinking of a covalent bond, right? Where you're actually, the electron spending more time over with another atom than with its atom, right? So in hydrogen bonding, what we're really talking about is an electrostatic interaction. You could have to draw things. I see Josie put, should we be prepared for drawing material? You could see that on the exam. Yeah, and there are some Gibbs free energy problems at the end of the chapter 2 questions. Sam said that, yes. So when you look at hydrogen bonding, what you're looking at for hydrogen bonding is electronegativity. So when we talk about water, if we use water as just an example, what's my electronegative part of water, right? I have, if my pen would work. It is not my technology day today, y'all. My Notability does not want to work with my pen. Showing it's connected, okay. There we go. Okay, so you have a water. What's electronegative inside the water? Anybody? Oxygen. Oxygen is electronegative. Okay, so if whatever atom it is that's electronegative, that's the one that's going to hog the electrons, right? So if you have water, you don't just have one molecule of water, right? We have a ton of water. So if you end up with the electronegative oxygen of one atom getting relatively close to now the slightly positively charged hydrogen, right? Because the electrons are being pulled to the oxygen. So the oxygen becomes partially negative. The hydrogen becomes partially positive. Now you have the same thing happening on the other oxygen atom and the other hydrogen atoms over here. Now you have a positive and a negative getting close to one another, right? That's your your electrostatic interaction. I know. Okay, so Megan says, I'm a little confused that the hydrogen bond is a non-covalent interaction. It is a non-covalent interaction because the bonds, the actual covalent bonds are within one water molecule. So I'm going to highlight them a different color. Okay, so I'm going to call this one is going to be blue, right? So I have a blue water and I have a red water. The bonds that exist, the covalent bonds are here and here. And we have on the other molecule, covalent bonds here and here. So those are the only covalent interactions that are here. So a hydrogen bond, this is why I hate the name hydrogen bond. A hydrogen bond is not really a bond. It's really an electrostatic interaction. And that's why a lot of times what you'll see hydrogen bonds drawn as are dots instead of lines. So those are the electrostatic interaction. That's a partially positive hanging out with a partially negative. So is that?
That makes sense. Thank you.
Sure. So whenever you're looking at this, now you have to think about what's a donor, what's a recipient, right? So whatever's has the hydrogen is the donor. So in this case, right, we have a hydrogen here. So this molecule becomes the donor. And this molecule has the oxygen. That one's going to be the recipient. Right? So this is why I say that hydrogen bonding is no fun, because you have to have a hydrogen, here's my hydrogen, bonded to an electronegative atom, and you have to have another electronegative atom for that hydrogen to interact with. So what electronegative atoms usually associate with hydrogen? Nitrogen, oxygen, and fluorine. So in this example, we're really only talking about oxygen, but it happens a lot with nitrogen and it happens a lot with fluorine. And it's because those elements are highly electronegative. Okay. Everybody okay on hydrogen bonding? If you have something, throw it up in the chat. Okay, so the next thing on my list was somebody asked about phospholipid flipping. So that was at the end of this chapter. Right on here. Right? Oop. So we know that phospholipids inside of a membrane are not stationary. we know that they move around a lot. So there are kind of 3 different ways that they can move around, right? If you have a phospholipid bilayer, looks like this, right? And then you have some like going back into the screen, and you have some coming towards you, right? Because it's 3-dimensional, and I can't draw that very well, but it is 3-dimensional. So lipid molecules can rotate. They can have rotational movement. So like if we pick this one, right, what would happen in a rotational movement is it would just kind of spin on its axis. Then you can have lateral movement where this phospholipid would go left or right or front and back. It can move in 3 dimensions while staying inside the layer. And this is really what contributes to the membrane fluidity model, right? And all of this lateral movement is really dependent upon all the different components of the lipid membrane. So do you have a whole lot of proteins there? How much cholesterol do you have? What's the temperature, right? All that kind of stuff. But at the same time, it's a bilayer, right? So you have another layer of phospholipids that are hanging out below. And so what this lipid can do, we'll do this one, what this lipid can do is it can actually flip-flop and change its orientation. So instead of being on the outside, now it's on the inside. And they call them, so flipases will take something from the outside to the inside, and flopases, I love the terminology, will flop them like this. So it's flip-flop, right? And so really the whole reason why you have all of this movement of phospholipids is so your phospholipid layer is not uniform, right? Because if your phospholipid bilayer is completely uniform, you can't do all the specialized things that you're supposed to do with the membrane. So flipping and spinning and laterally moving are all really important in keeping your phospholipid bilayer really diverse. And a lot of times it also has to do with charge, right? You're trying to keep a certain charge on the outside of the cell. And so different phospholipids have different charged head groups, so they can flip and flop and maintain whatever charge needs to be there. Okay, anybody else on lipids? Does that cover lipids? And anybody can put anything in the chat. I'm just going through a couple of questions that were given to me. Do we need to study classroom handouts, or were those just classroom? So the classroom handouts are ways that we use the material that we learned in lecture. So they're definitely fair game for the exam, because they're just illustrations of what we covered in class. So will the exam resources paper will be provided? Yes, it will be provided to you. You'll get an answer sheet, you'll get the exam resources, and you will get your exam. All right, is there a certain type of chapter questions we should practice? No, they're all good. There are regular review questions and there are challenge questions. They're all good. They're all good. I mean, I would focus more on the ones that cover what we discussed in class. You know, if you're like, oh, we like barely talked about that. Well, that's probably not going to be very heavily on the exam. So try to stick to the ones that we talked the most about in class. But if it's in the chapter, it's fair game. Okay. All right, the next one I have is the twist, DNA twist. Are y'all okay if I skip to that? Okay. If I don't hear a no, then I'm just going to skip to it. Okay, so that's chapter 3. That's chapter 3. Okay. So that was that was kind of early. Let's see. Slide. Here we go. So when you talk about twisting DNA, the first thing that you have to know is your DNA strand itself left-handed or right-handed? So there are 3 different forms of DNA, right? A, B, and Z. So which 2 are like each other? What do you notice? There are two, one of these does not belong. I feel like what was that on Sesame Street? I remember that right. So two of these look similar and one looks different. Which two are similar?
A and B.
A and B. A and B are similar, right? And how do I know that? Because if you look at A and B, look at the blue. Do you see how it's going in the same direction? Whereas in Z, the blue is going in a different direction. So what direction are they pointing to? Well, A and B are pointing to the right. This is right. And this one's pointing to the left. Right? Okay. Do we need to know how to find amino terminus and carboxy terminus? Yes, you do. You do need to know amino terminus and carboxy terminus. We covered that in class, and remember we drew some of a very short peptide, and we were able to see what was on the N terminus and what was on the C terminus. So the answer to Sam's is yes. Okay, so now that we know the inherent twist that's in DNA, now you got to think about DNA doesn't exist as a straight linear rope, right? What we're going to do in order to package it into our nucleus is to twist it on itself. So how do we twist it? So if you jump ahead a few slides here to supercoils, right? So here's our positive and negative, because there's a left and a right hand twist, and then there's a positive and a negative supercoil. So when you're looking at this, when you look at a positive supercoil, so A is an example of positive supercoil, what you're doing is you're twisting more to the right. So if DNA is right-handed, right? It's a right-handed twist. In a positive supercoil, what you're doing is you're compounding that right-handed twist. That's why we call it positive supercoiling, because DNA is right-handed, and then you twist in the right-handed direction. So what's happening when you do that is it's getting tighter and tighter. Have you ever taken a rubber band and just tightened it and tightened it and tightened it, right? So that's all you're doing. You're compounding that same twist. Now look at the next one where we have 3 negative supercoils. A negative supercoil is going in the opposite direction as the normal DNA twist. So a negative twist is left-handed. And that is going to actually straighten the DNA because you're going in the opposite direction than it's normally twisted in. So eventually you're going to straighten it. Now, what happens if I continue to introduce more left-handed twist? What is the DNA going to look like? Is it going to look straight still? No, it's not. It's going to start, if you take a rubber band, right, and doesn't matter which way you twist it, right-handed or left-handed, it's eventually going to coil up on itself. So the same thing is true about a positive and a negative supercoil. A little bit of negative supercoiling is going to straighten the DNA, and a lot of negative supercoiling is going to make it clump up again. And so that's what you see then. They call that knotting in negatively coiled supercoiled DNA. And that's what you see in that picture right there, is the knotting that happens. Okay, let's see. Oh, Sam says more with the 3D model. Not so much with the 3D model. We'll work with more of the 3D models, but that's a new thing that the homework system is doing, which I really love, because you can actually rotate it in 3 dimensions, and it's not just 2-dimensional. But I can't do 3D on the exam. It'd be cool, but I can't. So you're not going to do 3D manipulations on the exam. All right. Would you review? Yes. So somebody asked, review the main difference between the gene chip and the RNA-seq. And I actually have that on my list. So I'm going to do Cdna Library, and then I'm going to do that exact thing, Megan. I understand positive for the right hand and negative, but I thought that we had to follow the solid black arrow in the picture. Well, it's not following a solid or following a negative, because if you look at the arrows, they're both pointing, the solid black arrows both pointing in the right direction. It's what's overlapping, right? Is the gray arrow overlapping the black, or is the black overlapping the gray? So that's what's telling you if it's a positive or negative. But I'm not going to show you a picture of DNA and say, is this a positive supercoil or is this a negative supercoil? I'm not going to do that to you. You have to understand it, but you don't have to look at a picture, because how do you tell that? You can't really tell that. Okay. So Courtney, does that? And yes. Okay, good. Okay. So Cdna Library is what I have next. And that is still chapter 3, much further. Hold on. Let me let my puppy out. She's whining. Hold on. Okay. Y'all want to see her real quick? Come see Rosie.
Yes.
She's huge. Watch. Come see. Look. Come see. Let's see if I can tip it. There she is. She's listening to us talk science. She likes science. Okay. And Charles is here. You want to say hi? And then you got to go play your computer. Look. And there's Charles. Okay. Go play your computer, okay? No. All right. And son. Yes, we got puppy and son. All right. So I'm going to go down, I think, slide 59. Let's see. What's your question, bud? Right there. Go ahead. Okay, so Cdna libraries. So when you think about Cdna libraries, and it's the same thing with what y'all were asking me earlier about the gene chip and the RNA-seq, what you're starting with is mRNA. So when we talk about mRNA, if I want to know what the cell is expressing, what kind of proteins the cell is expressing. I'm back right here. Sorry, y'all. There you go.
Thank you.
You're welcome. Okay, so if you want to know what the cell is expressing, like what kind of proteins is this cell expressing? If this is a liver cell, it's going to have a certain subset of proteins that it's expressing. If this is a skin cell, it's going to have a certain subset of proteins that it's expressing. But sometimes it's really hard to say, is this protein there or not? But what's easy to say is the expression of this gene going up or down? And that's exactly what we're going to find out with a Cdna library. So we isolate all of the mRNA that's inside of a cell. And what we want to do is in some way, shape, or form quantify that. So in a Cdna library, you're going to get lots of mRNA molecules, right? And so you take that mRNA, and one thing that we know about mRNA is that mRNA has a polyadenylated tail. So if I design a primer that's going in the opposite direction, see how this is 5' to 3', my primer is 3' to 5', that has a poly T section to it. It's going to anneal to my mRNA. It will anneal to all the mRNA. Not just one, but every single one, because every single mRNA has a polyadenylated tail. So I'm only showing, in this figure, you're only showing one mRNA fragment, but there are thousands and thousands of mRNA fragments that are doing this at the same time. Okay, so you kind of have to remember that. Then you add an enzyme that will convert DNA to RNA, and that's called reverse transcriptase. In reverse transcriptase, it will amplify from that primer and make a copy of the mRNA. But that copy, the gray copy, is now DNA. And because it's a complement to mRNA, we call it cDNA, or complementary DNA. So basically we're going in reverse of the normal processes in the cell. So then what you do is you treat your sample with what's called ribonuclease, and the ribonuclease cuts the mRNA. Now it doesn't completely chew it up. It just partially degrades it. So if I partially degrade it, what I end up with is a cDNA strand with little pieces of RNA. Gosh, what does that sound familiar to? A primer on the DNA that's there for translation processes, right? So if I have DNA here and I have a primer every so often, if I just add a little bit of DNA polymerase, I can make the exact complement of my complementary DNA. So now I basically end up with, this is a double-stranded cDNA. Ho-ho, cDNA, right? But it's going to have, wherever that RNA was, the replication machinery will basically kick it out. So you'll end up with a little bitty gap in between where the primer was and the end of the last sequence. And so you need to fix those gaps, and you fix those gaps with DNA ligase. So now I have a perfect double-stranded cDNA. So now that I have that, I'm going to add some restriction enzyme sites to the end. And now that I have restriction enzymes, so these are RE sites, restrictin endonuclease sites, I can take that little piece and I can pop it into my vector. Remember in class the last time we were talking about vectors? You can just pop that right into your vector. And now I don't do that for just one, right? I have thousands. So I do this a bunch, and I get lots of plasmids. And I put these plasmids into different bacteria, and then I grow them on a plate. And I'm looking for that blue-white screening, and I'm going to put ampicillin or something like that in my plate as well. So if the plasmid is carrying an ampicillin-resistant gene, then the colony needs to grow on ampicillin, a plate with ampicillin in it, and you do that blue-white screening. Why did the ribonuclease need to partially degrade the mRNA? Because mRNA is really, really unstable. And so if you stopped here, pretty rapidly, you would lose that RNA. And so you couldn't easily make a copy of it, a DNA copy of it. So what you do is really quickly, you do the first steps really, really quickly, and you keep it cold so that you can get that nice DNA complementary double-stranded cDNA. Okay, good. All right, so it's the same kind of screening that you would in the generation of any plasmid. But okay, so what's the point? Why would I do this? Anybody? Like, I've generated this huge cDNA library. What do I do with it? It's a good question, right? Anybody? The whole point of this is to figure out what cell is expressing what protein. Can you compare it to other unknown? Well, you don't want to compare it to unknown. You want to look at those sequences, right? I can send this off to be sequenced. I could express it. I could do a couple of different things, but basically what I want to know is, this is my cell. What's it making? What kind of protein is it making? Or I could say, here's my cell. It's a cancer cell. I know cancer cells make X protein, right? So I should see the mRNA of it. I'm going to treat this cancer cell with drug X. And when I treat it with drug X, my expression level of my protein changes because I'm killing the cancer cell or whatever it is, right? So it's all about, cDNA libraries are all about what protein is being expressed and how can I use that information. Okay, so then we wanted to talk about the other kind of ways that you can look at expression. And there are two of them. There's gene chip technology, and there's next generation sequencing, or RNA-seq. It's the same thing, okay? So let's talk about both of them. Is that okay? This is really y'all review session. Okay, nobody's saying no. So gene chip technology is similar, right? Because we have a bunch of cells, right? And we isolate the mRNA, just like we isolated the mRNA in our cDNA library, right? Okay, we make our complementary DNA strand using the reverse transcriptase and that same primer. right? So I do the same thing. I end up with a cDNA double-stranded piece of DNA. The puppy's trying to get on the bed with Charles and it's not allowed. All right, so you make your cDNA double-stranded piece, right? Now, here's the difference. What I want a way to visually say this protein is being expressed. So what I have before I started this experiment is this chip. And this chip has little bitty fragments of DNA, single-stranded DNA that are attached, but they're not attached randomly. I know where every single one of these single-stranded fragments of DNA are, and these single-stranded DNA fragments correspond to genes, exons within the genome. So I can use this. I can say if something will bind to my chip, I can look at where it's binding and I can say, this is being expressed, this is not. This is being expressed, this is not. Just by looking at where on the chip something will bind. But I have to have a way to visualize that. So basically what I need is my binding DNA needs to be fluorescent. So how do I do that? Well, if we come back and we say, okay, I have this cDNA of the mRNA that's been expressed in my cell. And what I need to do is I need to label it with a fluorescent probe. And that's what the green ball there is the fluorescent probe. And so what you want to do for this is you want to convert it back into RNA so that you can actually label it. And so you put in RNA polymerase and you put in this fluorescent dinucleotides, these tagged nucleotides, and you PCR from your cDNA library using these fluorescent nucleotides. And now you have these little fragments that are fluorescent. And so we take all of those fluorescent little bitty fragments and we mix them with our chip. And the chip is really small. When I say a chip, y'all, I mean like it's really small. And so you load your sample onto the chip and then you put it into the machine, and the machine will look at that entire chip and it'll activate the fluorophore, that little circle, that little green circle. It'll activate it with a laser and it'll spread the laser over the whole chip. But only things that have that probe attached to it are going to light up. So only things where we have binding between the single-stranded DNA that were annealed to the chip, plus the little fragments here. Only if you have both will binding occur, and then you have a fluorescence occurring. If there's no binding, you get no fluorescence. And so I can say, okay, when I treat my cancer cell with X drug, the expression level of this gene goes down, this gene goes up. Yeah, it's not GFP, it's something different. It is something different, and there are lots that you can do, and that doesn't really matter what it is. But there's a problem with this. And the problem is where you start with that chip. Because if I say, okay, I have a liver cell, so I'm going to put only DNA sequences that are expressed in liver cells on the chip. Well, if it's a cancer cell and it starts expressing something that I didn't put on the chip, the level could go up, the level could go down, and I would never see it. So you start out what's called with bias, because what you put on the chip is what you know. But if something is changing that you don't even know is being expressed, you're going to miss it entirely.
So you can only see what you're looking for, basically.
You can only see what you're looking for. That's exactly right. You can only see what you're looking for. Now, it's quick, it's easy, it's cheap. So you can get a lot of information out of it, right? But typically, if you want really good science, you're going to do a chip, and guess what you're going to do? Next generation sequencing, or this is RNA-seq. RNA-seq. sequencing. It's the same thing. People call it different things. It's the same thing. Okay. So in RNA-seq, you do similar. You have your cells, you isolate your RNA. In this, it's very different from the beginning. You have these long strands of mRNA, right? It's the entire protein sequence. And what you do is you fragment it, you cut it up, and you add these little linkers to it. Charles, please don't do that. Come see Rosie. Okay, sorry, y'all. All right, so you have these fragments of your RNA. What you're gonna do is you're gonna convert it to cDNA. Now that they're little, you convert it to cDNA, and then instead of adding restriction enzymes to the end of it, what you're gonna do is you're gonna add what are called linkers. And those linkers are there not to cut anything, right? That's what restriction enzyme sites are for. These linkers are just there so that you can PCR the cDNA that was already there. So let's say that I started out with, let's just pretend, right? That there were 1, 2, 3 different mRNA sequences. So these are mRNAs. So you got 1, 2, 3 different ones. Well, it's really hard to detect. Rosie. She's stealing all my stuff. She's saying you're not paying attention. So you have 3 different mRNAs. If I only have 5 copies of this mRNA or 10 copies of this mRNA, it could have an effect on the cell, but I will never detect it because it's not enough of them. So what I need to do is I need to be able to amplify them. In order to amplify them, in order to amplify them, I have to add the linkers. So I add the linkers to it so I can go from having a couple of copies of each one to having an exponential number of those same mRNA copies, right? But however much I started out with, I'll have a proportional increase in those. So if I started out with a lot of this mRNA, I'm going to end up amplifying to make a ton. But if I start out with just a little bit, I'm going to amplify it and I'll end up with more to a detectable amount, but not that much more. Rosie is giving me a run for my money. Well, we usually go walking in the afternoon and we haven't done our walk yet. So I'm in trouble. It's my fault. It's totally my fault. So now that I've amplified them, I have enough that I can send it to be sequenced. So I go and I sequence every single fragment and I can tell, do I have 100 copies of this fragment? Do I have 1,000 copies of this fragment? And then I use a program. Come on, girl. I have however many copies, doesn't matter. I'm going to map those to each individual exon of all the different genes that they go to. So if I had 3 different mRNAs, it's going to map to 3 different genes. But it's only going to map to the exons of those genes because it's mRNA. So you're never going to see in RNA-seq a sequence for an intron because it doesn't exist in the mRNA. So this has no bias. Right? I'm not starting out knowing what I'm looking for. I get all of my products and I sequence it all and I map it to the entire genome. So I have no prior bias. But guess what what the problem with this is? Money, right? Sequencing costs money. So you try to do as little sequencing as possible. Right? But what good scientists do is they do both, right? They do both of the next generation sequencing and they do the gene chip arrays. And the question is, do the data back each other up? So for example, right, if we look at expression level of 2 different genes, gene X and gene Z, if I look at my array, my chip, it's the same thing as a chip. If I look here, this is an untreated sample. If I give this sample a drug, and I use the same method, and I say, okay, what happened to that transcript? What did it do? Go up or go down? Expression went down, right? Went from 1,849 to 825. So expression went down, okay? So my question is, if I use a different method that's unbiased, do I get the same result? So here's my RNA sequence. You get a different initial value, but that's okay. The question is, am I going to see the same trend? So I compare my treated to my untreated. What happens? It also goes down. So the two back each other up. I'm very happy as a scientist, right? Okay, the same thing, we can look at another gene. And I can say, okay, I'm going to first look at my chip, my gene chip array. I start out at whatever value it is. Untreated versus treated, what happened? Gosh, it's a little lower, but I would say that's not much of a change at all, right? So I would say unchanged. So then if I do the same thing and I look at my RNA-seq, I start at 421 and I go to 398. Basically the same. If you round those numbers, you're going to round them to the same thing. So basically that's unchanged. And the point is that the data agree with one another. And that's what makes saying, okay, now if I want to run this assay a bunch, I can just use the chip because it's cheaper and easier. And then I have this validation with this other experiment. All right, go ahead. Let's see.
I think you were mixing up the treated and untreated, but the same principle still applies.
Oh yes. Okay, I was.
And if
the treated
one
on the left, the untreated on the right
were to go down and the other not supported, then you could just think something might have gone wrong in the experiment.
Yeah, well, so if you do one experiment and the sample mRNA goes up, and you do the other experiment and the sample mRNA goes down, I would say something's really wrong, right? Maybe it's a bias in there. Maybe it's that you prepared the samples differently. I mean, there are a lot of questions to go through and trying to answer if you have that happening. I mean, this is golden, you know, this is beautiful data here, but this is textbook data. It doesn't always happen that way. Okay. Somebody else had something else. What else was on the list? What do we got? Charles, hold on one second. Charles, hey, I need you to take her out. Take her out.
Okay.
Thank you.
And keep her out.
Take her to the bathroom. Let her go out to the bathroom. Okay, so who else? Let's see. What else did we say? What other topics y'all want to talk about?
I talked about the Gibbs free energy calculation.
Okay, so
and I had like found one that was like in the homework for chapter 2.
On the online system?
Yes, ma'am. Mm-hmm.
Okay. Let's see if I can pull that up. I'll have to change my share. Hold on, let me pull up the homework system. Anybody else have anything else? Go ahead and drop it in the chat. Let's see. And I'm going to try to pull up our online book. Here we go. What problem was it in the homework? Oh, I gotta sign in. Tammy?
It was question number 6.
Number 6. Okay.
Mm-hmm.
Let me pull that up. Okay. So that was chapter 2.
Yes, ma'am.
Okay. Why? Whoops. The difference between standard and standard biological. So standard biological is what we're going to use while this is going. Standard biological is what we use basically as biochemists. Where did that go? Y'all, I'm trying. Here we go. No, that was the book. I want SmartWorks. So standard free energy are things like standard temperature, standard pressure, that kind of stuff, right? But you got to think in a biological system, there are going to be a few things that are different, especially concentrations of substrates and reactants, right? What do we assume everything is in just standard conditions?
Like constant pressure and temperature.
Well, constant pressure, constant temperature, but there's another one that's very different than a biological system.
Is it the 1 molar for the products
Yes.
and reactants?
Yes, 1 molar for products and reactants. You got to think inside of a cell, there's a reason why we don't want everything to be 1 molar, because there are lots of reactions that are non-spontaneous that won't go if everything's just 1 molar. So what we have to do is we have to change the concentration of the substrates and the reactants in order if we want the reaction to go in the forward direction or we want the reaction to go in the reverse direction. So you can't have 1 molar of everything. That's not like, it's not real world. Okay, so I have, it's a K equilibrium problem for number 6. Let me share this. Let me share this. So this is what I have for chapter 6. Tammy, is this the one you were thinking of?
Yes, ma'am. I said Gibbs free energy, but like that's, sorry.
That's okay. That's okay. I was just like, I must be on the wrong thing. All right. So if you're going to solve for K equilibrium, what do you have to do here? What's the, like, how do you know what formula to use? Where are you going to go?
Like KEQ is your products over your reactants.
Is your products of your reactants, but you're given delta H and you're given delta S. Right? And y'all know, have y'all noticed this on the homework system? Have you noticed this right here? Right? So remember there are relationships for the Gibbs free energy to K equilibrium and the other thermodynamic properties. So watch, if we, let me pull up the notes. If you go here, let's see. It's going back and forth. Let's see. That was pretty far. That was chapter 2, right? So you should have all your formulas here, right? Okay, so watch. I'm gonna switch back to my other screen, so y'all know which problem we're talking about now. See if this works. Okay, so here, if we want to know the relationship between delta G and K equilibrium, we're using, this is what I call the Ratlink equation. You remember that? So what we have to be able to relate, you have to be able to solve for delta G. So how are you going to solve for delta G? What are you going to do? Ooh, don't leave a meeting. You're going to have to do, let me write it. Delta G is equal to delta H minus T delta S. Remember this? Yes? I don't know where the picture went. Hold on. I lost everybody's picture. Okay. So once you use this formula, now what else do we know? We also know delta G is equal to negative RT ln K equilibrium. Right? So the first thing you do is you solve for delta G. Because you know delta H, you know delta S. What did they give in the problem? They gave delta H is negative 9.3 kilojoules per mole. Delta S is 75.5 joules per mole K. And I have 25 degrees Celsius. So what do I need to do?
Convert Celsius to Kelvin.
First thing is convert Celsius to Kelvin. Okay, so what do we get when we convert Celsius to Kelvin? 25 degrees Celsius is equal to, add
273.
So you get 298 Kelvin, right? Now, what's my other problem?
You need to convert either the kilojoules to joules or the joules to kilojoules.
Correct, correct. And what I would do is I would convert everything to kilojoules, because that's what we're going to assume is in our following equation. So I would convert everything to kilojoules. So that 75.5 joules, you need to convert that to kilojoules. Then you should end up with something that gives you a delta G of, I got negative 31.8 kilojoules per mole. Now you take that delta G and you put that into our second equation, right? And so we say negative 31.8 kilojoules per mole is equal to negative, what's R? What R are we going to use? We talked about this in class.
Is it the 80257?
8.314
8.314, okay.
joules per mole, okay? And then you're going to multiply times, I'm running out of room. I don't know where to put it. Times our temperature. We said our temperature was 298 ln of K equilibrium.
Can you
Right.
scroll down, please?
Oh, I'm sorry. Is that better? I think it's, there you go. So now you have to solve for K equilibrium, but notice that you have the natural log there. So what's the opposite function?
E.
E is the opposite function. Okay, Megan, you got a question?
Yes, ma'am. So isn't that R constant in joules, but we have delta G in kilojoules?
Kilojoules, yes. You're gonna have to convert.
Okay.
It doesn't matter, but you're gonna have to convert. Okay. So what do you get? It should be relatively big. Anybody? And let's see, while y'all are solving that, let me look at the questions. Can we go back and practice homeworks again? I don't think the system allows you to do that. Maybe it does. Y'all have to tell me. Can you go back in and see it? I think once you submit it, I don't think you can, but y'all will have to tell me. All right, when learning the math, should we focus on how the equations were created, or just focus on trying to solve them? Focus on trying to solve them. I think it's important for y'all to know where an equation comes from, so that it's not just like memorize this, memorize that. But I'm never going to ask you to derive an equation on an exam. I'm never going to ask you to do that. That's crazy. Do we have to know the individual structures of each nitrogen base? You should be able to pick them out. You should absolutely be able to pick them out. You don't have to draw them from scratch, but you do have to pick them out. And are there any equations that we have to memorize for the test? I'm giving you all of the equations that are on the inside cover of the textbook. So if you have a hard copy textbook, and I posted this on Moodle, but here, I can't see that here. Those are the equations that are posted. And if you go to Moodle and you look right up above exam 1, there should say something like exam resources, and you should see what it is. And that's what I printed out, and that's what you're going to have on the exam. So you only have to memorize things that are not on that sheet. So Okay, and Megan, you can see your homework questions?
Yes, ma'am.
Yay. Okay, so that answers that one. So you can go back and see them. So that's good. Okay, what else do we have? Anybody got an answer for the homework problem that we were solving? Should be big. Anybody? Yeah, that sounds about right. What are my units on that? Oh, it's an equilibrium. There's no units. Haha. A unit, a number without a unit is meaningless, but in this case, it's a unitless number. Okay. What are the questions? You got punked. I know, it's so terrible. All right, what else y'all got? Because we're coming up on an hour. So
for this problem, you don't plug anything in for KEQ, right? You just
you're solving for KEQ.
for KEQ. Oh, okay.
That's what you're solving for.
I think I like redid the equation wrong some type of way. I don't know. But thank you though.
It's okay, no problem. How many essay questions should you expect? Some? Some multiple choice, some fill in the blank, some problems, some essay. Just some of everything. More multiple choice than essay, I can tell you that.
I have a question.
Sure.
I don't want to sound slow, but how do you say, how do you change the kilojoules to joules?
Okay, so if you're in, let's say you have negative 31.8 kilojoules, right? So it's just dimensional analysis. So put what you want on the bottom and I'm sorry, put what you want to get rid of on the bottom so that they cancel, and then put what you want to get to on the top. So how many kilojoules are in a joule, or how many joules are in a kilojoule? That's what you have to know. So
I'm
there,
not sure.
kilo means 1,000. So there are 1,000 joules in 1 kilojoule.
Okay.
So it's all about those metric prefixes.
Okay, thank you.
Yeah. Blue-white screening. Blue-white screening is only used to determine which plasmids have actually picked up your gene of interest. That's correct. So if you have whatever PCR product, this is my PCR product, right? And what I do is I cut one end with one restriction enzyme. I'm just going to call it RE1. And then I take another one and I cut it here, RE2, right? What I want to do is I want to go into my vector. Here's my vector. And here's, let's pretend like right there is the multiple cloning site. What I want to do is I want to cut in the multiple cloning site with restriction enzyme 1 and also cut with restriction enzyme 2, so that I end up with basically a linearized piece of DNA that has a restriction enzyme site here and it has the other one here. So now what I do is I take this piece of DNA right here and I use ligase, and these two ends will ligate together and these will ligate together. And so I'll end up with a plasmid, right, that has those restriction sites, one here and one here. Whoops, if I pick the right color, maybe scroll up, right there. And then in between those two sites is now the PCR product, right? So my plasmid basically got bigger because it picked up the gene that I was trying to clone. Okay, so we good on that so far? So the question is, how can I pick plasmids that have the insert? So this is have, and then versus ones that don't, right? So what ends up happening is we take a gene, right? And we put part of it on one side and part of it on the other. And so if there's nothing in the multiple cloning site, this gene is basically good to go. It's called LAC-Z. LAC-Z, and LAC-Z codes for beta galactosidase. TOSID, beta galactosidase. So beta galactosidase is an enzyme that will convert X-gal. It'll actually cleave X-gal. And so if it can do that, your colonies will change color. So if I have the insert in, so if I come over here, I still have that X-gal gene. I have the LAC-Z gene there. And if the insert is in there, LAC-Z doesn't work, right? So LAC-Z does not work here, but LAC-Z does work here, right? So I can use that as a readout for whether or not my insert went in, right? Because if I have on the plasmid another gene, and this is like, let's say AMP-R. Well, I'm gonna have AMP-R on both plasmids, no matter what. So the colonies that grow on ampicillin plates have the vector, but now I need a differentiation. Is that vector a vector with an insert, or is that a vector without an insert? And so if you look, the white colonies are ones where it's not functional anymore. So we no longer have LAC-Z because the beta-galactosidase protein cannot cleave X-gal. And that's not so important, but it's important to know that you're going to have a color difference. So if the LAC-Z is active and it's still going, it can cleave it and you're going to get blue colonies. So it's all about, did the insert work? And if it worked, you basically disrupted a gene. And because you disrupted the gene, you're not making the protein, and therefore the protein can't do its job. And that's your readout for insertion or not into your multiple cloning site. Edward, does that answer your question? Let's get ready. Yes, no problem. No problem.
So if the insert works, then the protein can't do the job?
Yep. So
Okay.
protein non-functional is non-functional. Any other questions?
I'm sorry, you said if the LAC-Z works, then the protein is non-functional?
No. If LAC-Z is interrupted, so here's
Oh,
the case
okay.
here. If LAC-Z is interrupted, it won't express it. And if it doesn't express it, then the protein is non-functional, right? It can't do its job. Is CRISPR fair game? Yes, CRISPR is fair game. Absolutely. It's in the book. We spent a lot of time in class on it. Yeah, it's fair game. You know, I love it. Absolutely. Anything else?
So we can print out the formula sheet for the test?
No, I am going to provide it to you.
Okay.
So the formula sheet that's there, if you go to Moodle, you're going to see exam resources right above exam 1. And that's what it's the periodic table, it's the equation sheet. And then for a future exam, you need the codon chart. So I give you the codon chart, but you won't need it for this exam. Okay. Anything else?
Is there a way that we can have an extension on the homework and the quiz?
And the reading quiz for chapter 3?
Yes, ma'am.
Yeah, I could do that.
Okay.
I could
Just till like after the test or something like that.
Yeah, I can give you all a couple of more days on that.
Okay, thank you.
Yeah, I'll give you, what I'll do is I'll make it due next Monday. That way, if you're doing classwork during the week and that kind of stuff, you have the weekend to get it. How about that?
That works. Thank you.
Yeah. Yeah. Okay. Anybody else? Partial credit. Partial credit for what? For essay. Oh, I do partial credit on everything. If it's multiple choice, I can't give you partial credit. But if it's problems, if it's essay, what I try to give as much partial credit as possible. So don't leave anything blank on my exams, ever, never. Okay. because then I can't give you any credit.
Are you posting this recording on the Moodle page?
I'm going to right under where I announced that we were going to have this.
Thank you.
Mhm. Anybody else? Y'all can all laugh at seeing the puppy and Charles and
It's adorable.
I'm glad you think so. All right, anybody else, or I'm going to go ahead and sign off. I wish you all great luck studying. I'm sure you're going to do great.
Thank you.
And I'll make sure to bring my sunglasses tomorrow. All right, well, y'all have a good night, and I will post this. It's going to render, and then I have to post it to YouTube, so it's going to take a little bit, but I'm going to get started on it right now. I'll post it so it'll be available as soon as possible. All right, y'all have a good night.